everyone, and welcome to today's webinar, Scaling Up Organoid Research, a New Approach with Assay-Ready Organoids. I'm Cassie Saltman of Labroots, and I'll be your moderator for today's event. Today's educational web seminar is presented by Labroots and brought to you by Molecular Devices. To learn more, visit www.moleculardevices.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speakers, Angeline Lim, Senior Application Scientist at Molecular Devices, and Andy Bashford, 3D Biology Product Manager at Molecular Devices. Andy, you may now begin your portion of the presentation.
Hello, and thank you for joining our webinar today, where we're going to be talking a bit about scaling up organoid research and introduce a new way that molecular devices can help by providing assay-ready organoids. We have two speakers today, and we'll have a live Q&A session at the end. To introduce you to our speakers, my name is Andy Bashford, and I'm a 3D Biology Product Manager with Molecular Devices. And I've been with the company a number of years and originally started out with customers as a field-based application scientist with our high-content imaging systems. I'm also pleased to introduce Dr. Angeline Lim, who is one of our senior application scientists based out of our laboratories in San Jose, California. And Angeline is a very experienced scientist and works with all of our instrumentation to run experiments and collaborate with customers. So it's great that we can have her here speaking today. And for the agenda for the session, I'm going to talk briefly about some of the benefits and challenges of 3D biology and introduce some of the solutions that we have here at Molecular Devices. I'm also very excited to introduce our new service that we can offer to scale up organoid production for our customers. And then Angeline is going to talk more about the science and introduce two studies. One looking at some work to validate our bioreactor technology, which is used in the production process, with a specific example of patient-derived breast cancer organoids. And then she's going to show a nice example using these assay-ready vials in a colorectal cancer example with an automated workflow for a high-throughput screen. And so if we take a look at the bigger picture, Molecular Devices has a number of customers that are working in the drug discovery process, whether it's understanding fundamental cell biology, disease processes, or enabling screening for therapeutic interventions that will ultimately progress to the clinic. And this whole process is very costly and time-consuming, and it can take up to 20 years and billions of dollars to successfully develop a therapeutic from start to finish. And one of the many challenges that the industry faces in this process is that a high proportion of drug candidates fail in clinical trials. That's to say, they only fail once they've been administered to humans. And in part, this is because the models and tools that are used earlier in the process either do not fully replicate the true human state, either because they're too simple or there are too many species differences in the model used. And because of this, we're seeing scientists move more towards a greater use of 3D in vitro models. And this paradigm allows scientists to increase assay complexity compared to the more traditional 2D culture techniques. And by growing structures in 3D, we can allow more cell-cell interactions. If there's addition of an extracellular matrix component, this can support cell matrix interactions. Maybe there are other growth factors or cytokines or signaling cues or even biomechanical forces that can help to create a more accurate representation of the cellular niche. And this can all have a big impact on cellular behavior, maybe improving differentiation, the cellular phenotype, and expression of specific functional machinery. And ultimately, this can help in vitro models be more representative and improve their predictive power and help increase their translatability to the in vivo state. And another point here is that it can be used with patient-derived cells to help improve their relevance further. And when you compare it to in vivo animal models, these systems can be a bit easier to scale and comparatively quicker and cheaper. And this can allow more iteration and a more targeted approach to answer critical questions. And something that's really important is that it's about the right model for the right scientific question. We see customers working with a whole range of 3D models that vary in complexity, including spheroids grown maybe in round bottom plates that are maybe easier to start with, microtissues grown with extracellular matrix, hydrogels, or a mixture of more complex cell types. And looking more at complex models, there are stem cell derived organoids that can self-organize and start to recapitulate some of their innate developmental structure. Similar to this, there are tumor-like organoids, which model the tumor microenvironment. And we see other examples where scientists are really pushing the level of complexity with the use of organ-on-a-chip technology, maybe including microfluidics, pressure, stretch, or other organ interactions. And all of this is balanced about the investment of time and resources and expertise to get the appropriate answer from the system that you're working with. Yet, thinking about this, there are still a number of challenges that exist around 3D biology. And many of these mean that it's quite difficult to scale sufficient materials to work with your experiments. Firstly, the process is time consuming. It can require a lot of effort and a lot of time from highly skilled staff to grow these cultures. And even then, it can be quite operator dependent. Getting the complexity is one thing, but having consistency is also critical to help enable meaningful results. Some systems themselves lack robust methods, meaning that maybe slight differences in reagent composition or operator technique can lead to variations. And many systems also lack industry consensus, and so multiple protocols are being used or maybe even still developed for certain models. Automating the process can also be quite complex, with a number of challenging steps to optimize and certain processes requiring a high level of dexterity. And then once you have your model or you've completed your assay, getting the relevant data out at the end is also a real challenge. You've invested all this time and money into the complex model, and then it's critical that you get a value in terms of the data readouts, whether it's 3D analysis or detailed omics readouts. And more broadly as an industry, in order to overcome a number of these challenges and help scientists to better utilize these more complex 3D models, we believe that there are a number of actions that will help. And firstly, we think it's going to be very important to increase access to organoids, to lower the barriers to entry and enable a situation where these complex, more representative assays become the default day-to-day approach. And we think that automation will have a critical role here to allow scaling of assays in a very consistent way and help limit variation and facilitate some of the longer culture durations that are often required in these models. Also, utilizing artificial intelligence will play a key role in helping to allow the data to drive improvements, both during the culture process, but also for assay readouts. And finally, linking all of these processes together to create more end-to-end workflows that allow you to automate the full process, maybe with development of cell lines, culturing complex systems, and using AI-driven decision-making will help to achieve scale for higher throughput. And so molecular devices can help here in several ways. And if we break the process into 3 phases, developing the starting material, growing the complex 3D cellular model, and then getting a useful assay readout from them at the end. To start with, if we focus on the culture of the 3D models, and really the core of this webinar today, I'm really pleased to introduce our new organoid expansion services that we can offer to customers. And this is where we take your organoids, expand them in our proprietary bioreactor technology, and then send them back to you in an assay-ready form. And this allows you to remove the need for doing your own scale-up and simply have a stock of high-quality organoids that are ready to use when you're setting up your experiments. And I'm going to talk a bit more about how we do this later on. We also have our Organoid Innovation Center, which is a custom automation setup that we can build for customers. And we have one of these already set up as a showcase in our San Jose labs, which allows us to collaborate with customers and develop workflows. It also means that when we provide this custom automation platform to customers, we can really support them as a tried and tested solution. The next instrument, in partnership with Advanced Solutions, is the BioAssemblybot 400. And this is a 3D bioprinter that has a 6-axis robotic arm and interchangeable tools, or hands as we call them. And this allows you to have a wide range of printing functionalities, but it also enables you to dispense liquids and move plates and do much more with the system. It comes with dedicated design and automation software so that you can use the system to build 3D models and then combine it with maybe an automated incubator or a high-content imager to monitor assay growth, run assays, and facilitate the building of more complex workflows. And then finally, launching later this year, is the CellExpress AI. And this system will enable automation of seeding, feeding, and passaging of both 2D and 3D cell models. The software will use AI analysis tools to monitor cultures and make decisions about culture progression. And it can keep track of all elements of the culture and allow you to have much more control and consistency around how you're automating your assays. So keep an eye out for more information on this in the future. Now, if we briefly think about how molecular devices can help in a broader view, maybe earlier in the process, there may be a need for cell line development. Before you even get to growing up the cells, you might need to have some genetic editing of cell lines. And we have instrumentation that can help here too. We have the QPick series, which are a series of microbial colony pickers, which can help with creating and managing large genetic construct libraries, such as plasmids for gene editing. We also have the ClonePix mammalian colony picker, which can help both screening and selecting cellular clones, with stem cell specific pins, which can allow for the selection of cells with specific attributes. For example, say you wanted to select a GFP positive stem cell clone. And there are also tools to help with monoclonality. This can be the dispensing of single cells into wells using the DispensCell single cell dispenser, or imaging and monitoring to verify clonal populations using the Clone Select Imager. And this process can be critical in a regulated environment, especially when you need to confirm that genetically modified cells are all growing from a clonal source. And if we now look at the other end of the process, there are also tools that can be used from an assay readout perspective. Our SpectraMax range of single and multi-mode readers can cover fluorescence, luminescence, absorbance, and many other modalities. And a really common example of this in 3D biology might be using a luminescence-based ATP assay to look at cell viability. And these systems come with both acquisition and analysis software. They can be fully validated, and we have software solutions that can work in GXP environments too. We also have our Flipper Penta high throughput cellular screening system, which can use fluorescence, luminescence, and also has fluidic compound addition. And a nice case where we're seeing this in 3D biology is with cardiac organoids to track beating and calcium activity in a time lapse simultaneously across a whole plate of organoids. And then we also have our range of Image Express high content imaging systems, both wide field and confocal, that can allow you to monitor growth or complete more advanced optical sectioning with our Image Express confocal HTAI. And these come with analysis software that uses deep learning segmentation and machine learning classification to accurately quantify complex 3D structures and really get as much data out of these assays as possible. And Angeline is going to talk a bit more about these today with these imaging-based approaches using the assay-ready organoids in an automated workflow. And so to introduce our new organoid expansion services, this is something that we're really excited about, and we really think it provides a viable alternative to scale up of organoids in the laboratory. And when we think about the manual process of scaling sufficient organoid material to perform screening experiments, it requires a lot of steps of passaging and reseeding. And it would typically involve creating a lot of gel matrix domes in many wells across many plates. And this manual process can be quite time consuming and requires highly trained staff. And even then it can be quite prone to variability, which can lead to inconsistencies in organoid size or morphology, and ultimately mean that the process is very challenging and quite a big investment. And as an alternative to this, we can offer organoid expansion services where we take your organoids and expand them in our proprietary bioreactor technology and then provide them back to you in a frozen assay-ready format. And this technology can produce upwards of 6 million organoids in a single batch. And so let's say you're working with 96 well plates and you're using maybe 100 to 200 organoids per well. This means that you can work with hundreds of plates of data that all originate from the same expansion batch. And this means ultimately that you can have a stock of high quality, off-the-shelf organoids. And then when you're ready to begin your experiments, you can simply thaw them out, plate them, and get going without the need for any additional expansion. And now this is a bespoke service. So we consult with you to discuss your specific needs, and we can tailor the culture conditions to work with your organoid biology. We can discuss the scale of production you need, the number of organoids per vial, and we also offer a range of quality control options. This can be for more standard post-thaw viability or maybe STR profiling, or more in-depth confirmations such as response to specific compounds, whole exome or RNA sequencing to confirm that the bioprocess has maintained all of the essential characteristics of your organoids. And so how do we do this? The key is all thanks to the proprietary bioreactor technology. And the concept is that the bioreactor can grow organoids in a thin gel matrix layer and have a continuous media perfusion throughout the expansion. So this means that the culture is much more homogeneous with better access to nutrients compared to the gradients that you would see with dome culture, which can be dependent on dome size. It also means that there is no feast-famine cycle from the media, ensuring that there's always a supply of nutrients and removal of waste metabolites, helping to improve the growth consistency. And the scale of the reactor means that organoids can be grown in many more numbers per batch. So you're achieving a big increase in the scale, but they're all exposed to the same growth environment, providing a very consistent product. And with our current generation of bioreactor, we work with organoids that grow in a gel matrix. So they need to be in a position where they can be passaged, frozen, and shipped to us to continue the process. And we typically work with patient or animal-derived organoids, and these can be healthy organoids or diseased tissue. And they're typically also epithelial organoids, so common examples would be from the GI tract, breast, lung, ovarian, liver, pancreatic, or solid tumour. But if there's interest, then please do get in touch. We can always discuss your needs further. And so with that introduction, I'm going to hand over to Angeline, who can talk a bit more about the science and some of the case studies relating to the use of these organoids.
Oh, thank you, Andy, for the very nice overview of the organoid expansion technology. For the next part of the webinar, I will talk about two case studies. The first one is a study that looks at the bioreactor expanded organoids, and then these are compared to the manually expanded ones. In the second case study, I will show an application example that uses these organoids in a screening workflow. Going on to the first study, the main aim here is to validate the bioreactor expanded organoids. And in this example, we're going to show the validation study using patient-derived breast cancer organoids. To compare the organoids that were expanded using the two different approaches, first, organoids were seeded and cultured manually. They were then separated into two groups where they were either expanded manually, as at the top of the screen, or in the bioreactor, as you're seeing the workflow at the bottom of the screen. Organoids were grown for the same length of time and harvested using the same protocols. After harvesting, the organoids were then analyzed. And do note that for the bioreactor expanded organoids, they were harvested, frozen in cryovials, thawed before analysis. And we did this because we wanted to simulate what a user would be doing for the experiment. As part of the analysis package, we ran immunohistochemistry studies, whole exome sequencing, RNA-seq, and also a functional dose response study. So on to the immunohistochemical study. Biopsy sample was taken from a patient who has not received any treatment. And shown here on the left is a representative bright field image of the derived organoids, and this was grown in manual culture. So for this study, we looked for common markers that are related to breast cancer, and we wanted to see if these markers are retained in the organoids. Here in the middle is a table that summarizes either the presence or absence of common markers. And in summary, or basically what we see is that the organoids retain their original ER, estrogen receptor, progesterone receptor, and HER2 subtype status following expansion either in the bioreactor or manually cultured. And we also find similar results for the other markers that we looked at, such as P53 and the cytokeratin markers, namely CK14 and CK18. We also ran whole exome sequencing to check if the genetic profile of organoids changes between the two expansion methods. Here, specific driver genes were selected for investigation, and this list was actually based on the one that was previously described. The analysis indicated that organoids expanded using the two different approaches were similar. So essentially, organoids expanded in the bioreactor retained the same genetic profile as those that were grown or expanded in the manual culture. We ran RNA-seq analysis to see if there were any differences in gene expression levels in the bioreactor expanded organoids. Now, this was carried out in 3 independent studies. The analysis showed no difference in gene expression patterns between the two expansion methods, indicating that organoids cultured in the bioreactor does not significantly alter gene expression. For the last analysis, we did a dose response study. We carried out an 11-point compound titration, and these were performed in quadruplicates. So organoids were incubated with various compounds for 5 days, and this was followed by an endpoint cell titer glow cell viability assay. And so you're looking at the top 2 graphs here, and these shows the dose response curves for the effect of the compounds on breast cancer organoids. The data points in black are from manual cultured organoids, and in red are data points from the bioreactor grown organoids. This study was performed in triplicate, 3 independent triplicates. And so on the left is data from organoids that were treated with the mTOR inhibitor, and on the right is data from organoids treated with paclitaxel. And note that the dose response curves between either manual and bioreactor-grown organoids are very similar, and they are not significantly different. On the bottom, we summarize the data in a heat map that represents the calculated EC50 values for all the compounds that were tested. So further analysis indicated that the responses to these compounds were not significantly different between manual and the bioreactor-grown organoids. And so to conclude this section, what we've done is we compared bioreactor-grown versus manually grown organoids and found no detectable differences. The marker expressions remained the same, and so did the whole exome sequencing data and also gene expression patterns. And so this provides the assurance that the organoids that we grew in our bioreactor can be used for large-scale application studies, such as screens and assays, and I'll talk about such a case in the next section. So the second case study that I want to highlight is application-based. This proof of concept study looks at using patient-derived colorectal cancer organoids at scale for screening purposes. So a quick recap of the organoid culture and screen workflow. Andy has presented something similar previously, but here I would like to focus more on the timeline. Now, most people doing such a screen will first need to have sufficient quantities of organoids before setting up their screens. And typically, one would need to go through several rounds of organoid expansion, and this depends on the organoid line. The growth rate can vary. Some are a lot harder to expand, and some are relatively easy. But we'll just give it an estimate of about a 1 week expansion, 1 week for each passaging round. And so as an estimate from start to finish, you may take about 1.5 to 2 months before you can get your initial round of raw data. Now, if you have access to bioreactor expanded organoids, you would then follow the workflow that's on the bottom half of the screen. You would start with already sufficient quantities of frozen organoids. These are ready to be thawed, plated, and ready for your compound testing in 48 hours. So from start to finish, this screen will take you about 10 days. And so using pre-grown organoids, such as these in our bioreactor-grown organoids, leads to significant time savings, enabling you to get your results a lot faster. So here you can estimate it's about 5 to 6 times faster. And so back to the experiment. To set up this assay, we started with frozen vials of patient-derived CRC or colorectal cancer organoids. So you can see the workflow at the top of the screen. These organoids were thawed using standard protocols, and then they were mixed with Matrigel. We seeded approximately 150 to 200 organoids per well, and then we let the organoids recover for 48 hours before adding compounds. Now, in total, we tested 8 different compounds at 6 concentrations. We also ran this with the appropriate controls. After 5 days of compound exposure, we then ran various endpoint assays to assess compound effects. So here is a montage of images of the organoids after compound treatment. The organoids were stained with phalloidin, shown in the green channel, ethylium homodimer in red, and also Hoche, which is for nuclear, that's shown in blue. Now, while luminescence-based studies, such as the cell titer glow assay that I mentioned earlier, can give viability readouts, and that's based on the presence of metabolically active cells, the cell titer glow assay or luminescence-based assay is typically single parametric readout, and it's more suitable when you are doing high-throughput target-based screens. Now, with image-based assays, such as over here, you have the option to carry out high-content screening, which is a phenotypic-based approach. And generally here, you are focused on getting a comprehensive view of the system. You have the option and you can design your screen based on specific markers. Commonly used markers will inform you on the DNA content. Phalloidin, which as you can see over here in green, shows cytoskeletal structure. You can also add calcium AM to indicate viability. And actually in this case here, I use ethylium homodimer, which is a marker for dead cell. Depending on what you're looking for and how you design your assay, you can also use specific antibodies. Now, moreover, in image-based studies, you have the ability to get single-cell resolution. So this is particularly important when dealing with tumors due to their heterogeneous nature. And on the other hand, in addition, you can also, with image-based approach, distinguish compound effects on whether they are cytotoxic versus cytostatic effects. And I'm going to show an example here right now. On this slide, I have put together images from the same well over time. So each row represents a different compound treatment, and the top row here is the control, the meso control. Now going across from this in the screen from left to right, or the x-axis, that represents the days in culture. With the control group of organoids, you can see organoid growth over time. And in the second or the middle row, this is the 5-fluorouracil treated group. And you can still see that there is growth over time even after exposure to the compound. However, you can tell that even though the size of organoids continue to increase, the organoid structure looks different from the control above it. In fact, what it looks like here is that these organoids take on a more cystic and hollow-looking appearance. In the bottom row, these are organoids exposed to doxorubicin. And I think here is a clear example of cytostatic effects of the compound on organoid growth, where the organoids show growth arrest. Here is a time-lapse playback of the organoid CRC organoid growth. To quantify the observations, we developed a deep learning-based segmentation model to help us identify each organoid that you see in this image. And from that image segmentation, we can derive measurements related to the size of the organoid. And shown here is a plot of that on the right. Each point represents an organoid, and on the x-axis, it's over time. You can see over time the sizes on average increases. And this is what the same plot looks like for all the other treated organoids. From these plots, we can determine that compounds such as trametinib, romidapsin, and adavorcitab, and also doxorubicin inhibits the growth of CRC organoids. And so at the end of 5 days of compound treatment, we ran an assay to determine viability. Organoids were stained with calcium AM, a marker for live cells, and ethylene homodimer, which is a marker for dead cells, which is in red. So here is a panel of representative images that were acquired on the ImageXpress confocal imager. And similar to the previous analysis, we use the same deep learning segmentation model to identify all organoids and measure the intensities from the green and red channel. And actually here at the bottom, you can see the bright field image with the magenta pink outline around each organoid. This is the output from the deep learning-based segmentation. And so from the segmentation, we then extract or obtain the intensities from the green and red channel. And then the average intensity from the red channel is expressed as a ratio to the green channel. And in this way, high values indicate more dead cells per organoid. And this is shown in the graph on the lower right. Here, the set of data on the left are organoids that were treated with low concentration of the indicated compounds on the x-axis. On the right, we have organoids that were treated with high concentration of the compounds. And we find here that doxorubicin has significantly higher amount of cell death compared to the controls. So putting the two sets of data together, we find here that most of the compounds we tested, especially those at high doses, show mainly cytostatic effects. And however, only doxorubicin shows both cytostatic and cytotoxic effects. So sorry, just to orient you on the different graphs. On the left, it's the graph that shows the change in area, so that's going to tell us cytostatic effects. And on the right, that's the graph that shows the viability assay that represents cytotoxic effects. Now recall the earlier point I made on why one would take an image-based approach in screening. I mentioned the ability to do phenotypic-based high-content screens. And in this image, again, which is a montage of organoids exposed to various compounds, the red channel represents mitochondria. And so from this slide alone, I hope you can appreciate the phenotypic diversity that's present. And in the last couple of slides, I'm just going to give a very quick snapshot of the type of data analysis that can be done with such organoid screens. And then I will end up with a quick overview of automation solutions. One of the features with multi-parametric phenotypic readouts is that you will be swamped by lots of data. And so here is an example of a screenshot of one of my data tables. High content screening can give you lots of information, and if you were imaging your organoids at high magnification, you would be able to at least to some extent extract single cell features to describe the observed phenotypes. For example, here, for each organoid, I'm able to obtain 118 measurements that pertain to these different classes of measurements. So spatial information, the intensity of the various fluorescent channels, the positions of where they are, relative position, the texture measurements, and even the co-localization. And so all these measurements were put through a data analytics pipeline that includes data normalization, feature reduction using principal component analysis, or over here I'm later representing the data points in a UMAP. Now all measured features are represented as a phenotypic distance score shown here. And so to sort of explain this a little bit, the positive control is phenotypically distant from the negative control. And so that's represented by the orange bars of positive control, and here are negative controls. Here is another representation of a data set in a UMAP. Again, the positive controls are the ones that's indicated with the orange dots, and that's on the left. And on the right, that's where the negative controls are. Now, what I want to look at here is when I add the organoids exposed to doxorubicin, there, and this is at various concentrations, you can see there's a spread from the left to the right of where these treatment groups fall within this space. And now if we represent concentration also as a heat map, and that's on the right here, you can see that the high doses, compounds at high doses tend to fall on the left side of the chart, and the low dose compounds are on the right side of the chart. Furthermore, we can also use this to carry out phenotypic classification, where similar, so-called similar looking or phenotypically similar organoids are clustered together. And this approach is particularly helpful when one is trying to get a handle on what are the possible mechanism of actions of an unknown compound. So back to the slide that Andy showed earlier, we started by highlighting the challenges associated with 3D biology, especially at scale. And we have shown that our proprietary organoid expansion service helps to get you started by providing you with organoid in large quantities. But to get to do the screener assay at scale, you probably need an automation solution to help you with the throughput. And so here's a slide that shows the automation for 3D cell culture that we have set up in the lab. This is in our lab in San Jose. Our custom solutions team has worked with various customers. Is this playing? Yeah. Has worked with various customers to set up to customize their automation setup for their assay requirements. We can schedule different events to carry out automated cell culture process, tasks such as media change, such as what you just saw, it's a media change routine, and also routine imaging of your organoids can be accomplished on this automation system. So to briefly summarize, we believe that organoid technology holds significant promise in transforming drug discovery. However, there are barriers that still exist. And so here at Molecular Devices, we are helping our customers get over these barriers or challenges by first providing access to large quantities of your own assay-ready organoids. And the combination of the readily available organoids coupled with our automation expertise will definitely help drive your research and drug discovery efforts. And the last thing I want to mention, and that's this little image over here. Here's a sneak preview into what is coming next. We will be launching a new product that is made for automated cell culture, and specifically it is made to handle 3D cell cultures. And so do keep a lookout for it. More details to come over the next few months. And I'm going to post some information here. If anyone in the audience is interested in our organoid expansion service, please reach out to either Emma, who is based in Europe, or Erin Reisinger, who is based in North America. And with that, Andy and I will be happy to take any questions that you have.
Thank you, Angeline and Andy, for your informative presentations. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the bottom left of your screen and click Submit. We'll answer as many of your questions as we have time for today. Let's go ahead and get started. Our first question says, what is the scale of bioreactor used to grow organoids?
So I guess I'm happy to take this, Angeline. In terms of scale, I guess physically, you can think of it as maybe the size of your hand. In terms of scale from how many organoids can it work with, it depends a little bit on the organoid in question. Typically, it's between maybe 2 to 6 million organoids per bioreactor. But the team can also
stack up multiple bioreactors. So depending on the scale, depending on the needs, it can really be as big as necessary to fulfill the project in question.
Great, thank you. Our next question that we have here asks, do you store the expanded organoids for customers?
Again, I think I'll take this one. So in terms of the services we provide, the key thing really is that it's a scale-up service. So customers will provide an organoid to us, we'll scale it up, and then provide that back to the customer. With that in mind, the vials are frozen and the projects are very custom. So if there's a level of needing to make sure we hold on to sufficient vials to ensure their security for the duration of a project, that's something we can discuss. The other thing to think about as well is that today we discussed predominantly the bioreactor expansion, so very large scale production. But as part of that process, we also develop master and working bank stocks. And so that can be an important part of the storage of organoid lines. So again, if that's of interest, get in touch, and that's something we can discuss further as custom projects.
Great, thank you. Our next question here asks, how do you quantitate organoids?
Okay, I think I should take that one. Well, it depends on the context. If you're referring to counting the number of organoids in your assay plate, I use an image-based approach to count the number of organoids that's in our assay plates. If we're talking about how many organoids for seeding, that's a little bit different. I started with the organoid expansion type of vials, and then those vials are delivered with a fixed number of organoids per vial, so you can calculate your seeding density in this way. Other ways of quantifying organoids is by doing something like an ATP assay, which you can correlate the amount of ATP to the number of organoids in your culture.
Fantastic, thank you so much. Our next question that we have says, does MolDev provide models for AI analysis?
Okay, I'll take this one too. The Encarta image analysis software comes with a couple of base models that you can use to start training your specific model. So there's one for nuclei, and also one for cell, and I believe there's one for cell in transmitted light. And usually when I start with my analysis, I'll use one of these base models, pick the one that's closest to what I want, and then retrain based on that model.
Great, thank you. Our next question might be multi-part, so bear with me. It says CNS applications, neurons, and blood-brain barrier, blood-CSF barrier.
Happy to at least speak to it a little bit. I think the key thing there for me is at least it's speaking to neural type applications. And so currently from an organoid expansion service, that isn't something we're offering today, but it is something in terms of our other systems that can certainly automate the culture of those types of biology. And arguably that's almost more useful in terms of the duration that some of these models might need to be cultured for. So having that as an automated process might be quite helpful. And so, yeah, certainly something we're paying attention to for scaling up neural type applications.
Okay, thank you. Our next question that we have here says, for organoid imaging, is this done live or fixed?
For the organoid imaging, for the viability assay, this was done with live samples. Also for the bright field monitoring kind of imaging, that was done with live samples. back to the viability assay, I use a live dead kit, and so you use calcium AM that would label live cells, and that's a membrane permeable dye, and it only fluoresces in the presence of an esterase that's in the live cells. The dead cell marker is a deuterium homodimer, and that would only enter the cell when the membrane is compromised. You can also image organoids in fixed samples, and so I do that sometimes. I would fix the organoids and then stain with something like phalloidin just to show the actin organization within the organoids. So you could do either.
Wonderful. Thank you. All right, it looks like we have one more question here in the queue. So if you have any last minute questions that you'd like to ask our speakers during the live portion of the Q&A, please do so now. Type them into the Ask a Question box on the lower left and click Submit. I'll go ahead and ask this last one here. This question asks, is your automation system capable of doing cell or organoid passaging?
So I can take that. I guess short answer, yes. And so that touches on the Cell Express AI system that both Angeline and myself mentioned that has very recently launched. And this system really can automate the process of culturing stem cells, 2D biology, also 3D biology, whether it be spheroids or organoids. To answer the question more directly about organoid passaging, again, yes, and I know a lot of work has gone into working with that, whether it be breaking matrigel domes, seeding matrigel domes, and really trying to optimize that process. And I guess the key thing with doing that is it's trying to take out the operator variability. So by having an automated process for that, you can have a lot more confidence in how reliable that's going to be in terms of ultimately making the culture more consistent.
I can add on a little bit to that too. So we designed this system to handle both 2D cell lines and iPSCs and also 3D sort of organoids and spheroids. And so in terms of passaging, we can carry that of iPSC cells. And then for 3D, it would be for organoids that are grown in some kind of extracellular matrix. So something like Cultrex or matrigel. So basically the system is able to retrieve all the organoids from these domes, fragment them, and then resuspend them in more matrigel for reseeding. So this will help you to set up your assay plate, for example.
Great, thank you. We had a couple more questions come in, so I'll ask the next one here. This one states toxicology applications, hepatotoxicity, renal toxicity assessments.
So I guess in terms of toxicology, I guess that's something that we're certainly seeing customers have a lot of interest in, particularly from, say, GI toxicity. And so I think that's something to highlight, that we do sell, or we do work with healthy models. And so to have that as a means to then see maybe off-target toxicity, things like that. Angeline, not sure if there's anything else to add in addition to this.
Yeah, I'm not clear on whether this is with the expansion servers or or it's an assay type question. I don't have anything to add.
Not a problem. And we can always get an expansion from that registrant to fill in the blanks a little bit, and we'll answer that offline as well. So we have another one here that asks, have you correlated your organoid analysis with patient response in any case studies?
I would say no, we have not. We would love to, but just where we are, we don't have access to patient case studies to help us with what we do in the lab. Andy, do you have any examples or anything to add?
Nothing to add from a data side of things, but I guess
Mm-hmm.
always looking to collaborate, I guess. So if there are people out there that have access to that or want to partner and work together, I think that's something we'd certainly be interested to learn more about and see how we can work together with the partners.
Wonderful. Always
Absolutely.
growing and expanding. All right. And it looks like those are all the questions that we have at the moment. So thank you, Angeline and Andy, again. I do want to ask before we go, Angeline, do you have any final comments for our audience?
No, not from me.
Okay, perfect. Andy, did you have any final comments?
Only say I hope the webinar was informative, and yeah, to get in touch or to go onto the Molecular Devices website if you've got any further questions.
Wonderful. All right. Thank you. Thank you again, both of you, for your time today and your important research. We would also like to thank Labroots and our sponsor, Molecular Devices, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. Labroots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.